Hello, we are Gutcha Microsystems. I'm Lee. Kendall.
I'm Pedro. I'm Uriel.
And this is our project.
Okay, so as we all know, organ-on-a-chip technology is a revolutionary technology, and it lets us take human cells and grow them in vitro, and let us model actual human organs. And therefore, we don't have to test drugs on animals anymore, and we can have more accurate models of human cells and how they behave. In this class, we learn about a lot of the applications of organ-on-a-chip devices, particularly gut-on-a-chip devices, which are very good at modeling the intestine. And some of them are seeing how different organs interact with each other. The gut-on-a-chip can be used to diagnose diseases like inflammatory bowel disease, and it can also be used to see how viruses affect different organs. So for our project, we decided to focus on what comes at the end of any experiment, which is the imaging and the observation. So we know that the accuracy of any data that we collect at the end of an experiment depends on the quality of the observations we can make. And in this case, it would be seeing our cells under a microscope. Normally, we use a light microscope to get 2D images of the cells, and confocal microscopy can be used for 3D images. In the top, we can see like the traditional structure of a microfluidic device, in which there are two channels stacked on top of each other, and the membrane is horizontal. And the cells interact at the membrane. However, it's a bit difficult to visualize that using a light microscope, using a confocal microscope. And so we decided to see if we can come up with a device that bypasses that lets us see the cells interacting at the membrane using a confocal microscope, and we thought we might be able to do this using a vertical membrane. Here's an image of a normal 2D image using a light microscope, and on that side we can see a 3D confocal image, which is obviously more preferable to see how well your cells are doing.
Yeah, so just to recap, microfluidic models are only as good as our ability to make observations and measurements from them. So we believe that if we increase the quality of the observations that we make from these devices, we can make better conclusions and therefore lead to more effective experiments. So our research question was, how do we engineer a vertical membrane into a microfluidic device to obtain a higher quality observations at the end. So in sort of answering this question, we took a four-step design process, which started with the ideation step. We brainstormed ideas, and we sort of developed CADs for the designs that we had in mind. After developing CADs and assessing the different models, we went on to prototyping. The way in which we prototyped our devices was by simply 3D printing a mold and making the PDMS device in our mold. And then we put our devices together, we bonded our devices together via plasma treatment. After we made our devices, we had to evaluate the integrity of these devices. The main metric we used to evaluate our devices was leakage, because since the main point our device was trying to prove is that we could make a vertical membrane PDMS device, we decided to use leakage as a metric. So if there was leakage, we knew that the junctions in between the membrane and the PDMS device were not sealed. So after going through this process, we eventually came up with one design that was leak-proof. However, we did not get around to testing cells.
So to go into a little more detail about our device, our first idea initially was to have this mold, which essentially would print out, or that you could cast PDMS into it as this, showing that the channels would be on the top and the two PDMS pieces would be separate. That way you could plasma bond them with the membrane in the middle.
Can you guys point so we know what you're talking about?
Yes, sorry. So right here, you would put the membrane across this vertically and bond them vertically with using plasma treatment. And later on, close the channels with the top piece. However, we came up with a few issues right away when we tested this. One was there was no way for the membrane to actually be placed. While the membrane could be placed in the slit in between the two pieces here, because the top channels were open, there was nowhere for the membrane to actually bond to the top. So again, we would have to plasma treat 2 times, one to bond the 2 pieces of PDMS together, and one to bond either a glass slide or another piece of PDMS on the top to seal off the channels. But again, because the membrane would be stacked vertically, there was no way for that to adhere to the top piece, and therefore we had a lot of leakage. So our second iteration tried to fix a few of these problems. We instead came up with this idea of adding a slit in which we would add something that we were inspired from for past research. And this is a membrane holder. So essentially you would have 3 types of materials. One is a polycarbonate sheet that would essentially hold the membrane in place, and then an adhesive sheet in the middle to to adhere both pieces together. You can see here with the dotted lines that the polycarbonate sheet was not a fully rectangular sheet, but rather kind of an upside down U that held the membrane in place. And so this membrane holder would actually be placed in the slit, because the polycarbonate was an upside down U, it would allow for the membrane to still be intact on both sides of the channel. This device also came up with a few issues that I'll describe now. So one, there was still leakage, and we had a few days to figure out why. But eventually we realized that because the membrane on one side of the channels was exposed, when we would actually try to place the membrane holder inside of the slit, the membrane integrity would most likely fail because we were touching the membrane as we were placing it. And so this might have caused a lot of the leakage, but also the fact that this bottom part of the membrane was still exposed, and there was, again, no way for it to adhere to the bottom of the slit. And so this might have caused more leakage. A smaller issue that we saw was that there was no way for us to line up the inlets and outlets to the channels because we had no way of actually telling where they should line up when we plasma treated it. And so in order to fix that, this is our finalized device for the semester. So the only real change we made to the mold, at least, was add the 2 little crosses here that you can see in the actual device that would help us align the 2 PDMS pieces together, as well as removing the pre-cut holes that way we could cut them out later. But we also changed I guess the main change here for the device was essentially varying the membrane holder a little bit. So instead, now we would have 2 polycarbonate sheets on either side of the membrane, 2 adhesive sheets holding the membrane and the 2 polycarbonate sheets together. This did not fix the issue of leakage here because, again, the membrane was still exposed at the bottom, which meant there was no way for it to adhere to the bottom of the slit here. And so what we decided to do was add silicone paste to both the bottom and top of the membrane holder. That way, the membrane would adhere to the slit. And I guess to go into a little bit more on why we use silicone paste. Silicone paste is essentially PDMS. However, it's different than the PDMS we normally use because it does not need to, it can cure at room temperature. So meaning that you would not need to place the membrane holder inside the slit before putting it in the oven. Therefore, you could add the membrane holder afterwards and still have it bond to the slit. And again, this is where the membrane would go, and these are the dimensions for our device. And again, just to recap the problems we solved using this change membrane holder. It was quicker to insert because we didn't have to hold the membrane or the holder on both sides with an exposed membrane. The silicone paste covered the leakage. Similar to our past iteration, the U-shaped polycarbonate sheet held the membrane in place and made sure it was sturdy. And then we could later on punch the holes in the PDMS device after plasma treating it.
So using the finalized design, we assembled the device in 2 ways. One was the control, where we had the regular device, but with only, instead of the membrane coater, we only used a polycarbonate sheet. So this was kind of a proof of concept where we used the silicone paste, and as you can see, it did not leak the green food dye. And we also assembled with the actual membrane, and it also did not leak. So with this dye test, we showed that we successfully created a vertical membrane that could that would not leak and could be used for better imaging.
Yeah, so after we were able to figure out how to integrate the vertical membrane into our device, we tried to do cell studies. So we seeded cells and we stained them for cell viability assays. However, we were able to successfully seed the cells into the device, and then we were able to determine cell viability, but we weren't able to image the actual membrane, and we believe this was due to a couple of reasons. The first one being the PDMS itself. So the PDMS the bottom half of our device was very thick, and so this made it particularly difficult to image. So that forced us to use a lower resolution objective in the confocal. And additionally, the silicone paste that we use to seal off the junctions in between the membrane and the PDMS also may have caused some scattering of light, which led to lower resolution image and impaired us from actually seeing the membrane in the confocal.
To conclude, we learned 2 main things about the field of microfluidic devices. The first being that there is a lot of flexibility and a myriad of approaches towards the devices, which is exemplified by every approach that our class took. Each one was innovative and different. And this has to do with the field being relatively new. So like, in the past couple of years, there are methods which have been developed, but there's still a lot to be improved upon and perfected, standardized, or scaled up to make either more manufacturable or more readily available. Another thing which we learned about the microfluidic field in general is that there's a lot of potential for each of the devices, both from drug screening and testing, with a lot of different materials and the problems that come with each of those things. For example, the drug absorption, and also like the potential for the field to help with like better imaging and models for the human bio and the human as an organism itself. Because like as stated previously, animal models aren't the most accurate portrayals of the human body. Should we be able to talk for ourselves a few months ago, we would tell ourselves 2 things. The first is things will take longer than expected, especially with dealing with cells for a lot of us, this being a multidisciplinary project. It was an experience to be able to take care of like 1,000 little living things. You had to feed them, you had to change out the class when they got too full and like split them, you had to check up on them, and there would be little things where you just felt like a really bad parent, like, oh no, I forgot to like put a humidifier in there to keep them alive. And so just knowing that science, especially involving living things, will take patience and a lot of time, and that failure will occur. When we were putting the device together and putting like the membranes, we got to a point where it became almost routine, like, oh, you do this, you do this, you do this, and like we'll cut it. But even though we had like kind of a routine down, like sticking things together and creating the membranes themselves and then putting them in, it would be like out of 10 that we would make 1 of them might work. and then you put them in and it's not until you run it that you see whether or not it happened. And this is during the final stages of our device. Like prior there were a couple of other problems. Like we would tell ourselves that failure is almost to be expected, but it's not a problem. You just kind of have to keep pushing through. And one of the other things we learned in this path is like you might have to change things up. So there was a point during this project in which we're like, oh, maybe we have to change from the membrane and just like continue creating cells and like split the group sort of into two paths and see which one works. But then eventually like realize that, oh, the membrane can work, so then we just like reconverge back towards that ideal. But just being flexible. And should we have 3 more months, we would further test the devices with cells, try to improve imaging by either changing the device thickness, just to make it more visible with the vertical membrane. We sort of got some cells in to one of the membrane panels, but we would try to add a